Thank you, Professor. It's really a pleasure. It's a daunting task. Raja Hajar is clearly one of the world's leaders in gene therapy and has been at the forefront of bringing this strategy forward into clinical trials and has done almost all of the preclinical work to support this. And this was the provocative title that Professor charged with, are we ready to go in the clinic? And what I'll try and do is to summarize what we know, where we are in the field of both gene and stem cell therapy for particularly the area of heart failure. Reminding you that the concept that we're working is regeneration, and I think this topic really describes the ability to try and repair or recover damaged tissue, organs, or blood vessels. That's a rather daunting task, but as I'll say in the end, there are, in the United States alone, 4,500 clinical trials ongoing right now in stem and/or gene therapy in the attempt of regenerative medicine. So what we're looking at in the area of cardiovascular application, and particularly this morning in heart failure, is a reparative mechanism, which is the fundamental biology that the body uses, whether it's a large laceration or pneumonia or what other chronic diseases, tries to mobilize them. And as one person said, disease is a manifestation of the failure of stem cells. But I think that the topic will be somewhat controversial, but I hope to engage in questions at the end. This rather simple cartoon really describes the concept of using targeted gene therapy. It's very impressive as we talk about stem cells to recognize that they don't stay in their implanted site, particularly in the heart, for longer than days. And so the fundamental response is to trigger the heart's native repair mechanism, and whether that's entirely at the direction of specific upstream, if you will, targeted genes, the concept of maybe we don't need a stem cell, we would just use those specific genes to turn on that repair mechanism is quite attractive. This is a immunohistochemical study that really captures the concept of the downregulation that is well described in the setting of heart failure of critical genes. This is dystrophin, and this is in the context of a left ventricular assist device which completely unloads the ventricle. And as you can see on the left-hand panel, this is a control without heart failure. You can see almost total loss of expression of this critical gene in cardiac contractility in advanced heart failure at the time the left ventricular assist device went in. And here we are at about 3 months after LV support with the LVAD, and you can see that we can really upregulate this expression. And so it's this center panel that really makes the case for potentially being able to upregulate this without a surgical intervention. As I said, Raja Hajar, Mark Penn, and others have worked on the concept of identifying the molecular mechanisms involved in heart failure and potentially in recovery. And both using knockin and knockout models have confirmed a number of genes that are of critical importance. And the hypothesis of this is, as I suggested, that if we used a large volume of the gene and deliver it directly into the myocardium, could we really upregulate the body's native repair? And being that these are human recombinant genes, they offer no foreign protein to have any adverse response. Of the three things that are required to deliver gene therapy, the first of which was to identify a vector, which is very influential in the efficiency and the level and the duration of the effect of the targeted gene being instilled into the cardiac tissue. There are three options that have been described, and that is a virus or non-viral plasmids or liposomes. And the original application here was the use of viruses, and a number of viruses have been explored, and we'll talk about the use of a plasmid. The second really important component of requiring for gene therapy is a delivery method that will allow the gene to be into the target tissue, and then obviously the critical gene of interest that has a proven track record of being a critical gene in modifying heart failure. And AAV1 was the first cardiac gene vector used. I think many of you are aware of the introduction of clinical gene therapy. It was a field that was moving rather rapidly, and the approach initially was to find monogenomic genes or diseases for which you could theoretically do gene transfer and upregulate the expression and eliminate that clinical problem. The first virus that was used was adenovirus 1 serotype, and this is a very common exposure virus related to the common cold, but in the laboratory is one of the viruses that's used to induce myocarditis and a form of heart failure. This gene was coupled to the adenovirus and instilled in this young man with this hematologic clotting disorder, and unfortunately about 5 or 6 days after treatment, fulminant myocarditis ensued and the patient died. And this tragic event really put a halt to the area of gene therapy and has been one of the sobering aspects of, as we move forward into stem cell therapy, of saying, do we have the mechanism, we know where we want to be, but I think that this was really related to the vector and not really well defined on the front end. So when you look at the targets that have been explored to date, a lot of the focus and initial focus was certainly on calcium handling proteins, and again, this is Roger Jar's work. Beta-adrenergic signaling is another obvious component of contractility and effective therapy in heart failure. And I'll mention a few other genes whose role is also in stem cell mobilization. This outline of the sarcoplasmic reticulum is really particularly related to the concept of using the number one protein gating calcium flux in the sarcoplasmic reticulum and release for contraction is the sarcoplasmic reticulum calcium ATPase, or abbreviated as SERCA2A. This gene is really particularly influential and has been one of the genes most dramatically downregulated and similarly heart failure rescued by upregulation and gene therapy with this specific gene. So it has a very clear preclinical basis for use. However, there are other components, including phospholamban, and the relationship between SERCA2A and phospholamban levels really potentially make this a secondary target for gene therapy going forward. But this is the gene of interest in the first clinical trial. This went to a clinical trial in Europe and it proved a CE mark. It's a pretty standard phase 2A trial in which there was a dose ranging of low, medium, and high compared to placebo, and then followed up at 6 and 12 months. This graph really portrays the composite endpoint, which was worsening heart failure, need for a ventricular assist device, transplant, or death. So a fairly aggressive endpoint of clinical significance. And I think you can see very clearly that any of the 3 doses of the SERCA2A gene, referred to as MidCAR, was significantly better than the placebo group at all time points after the first 60 days. And this data was, although the p-value in the lower dose was not as robust as was seen in these 2 medium and high doses, it certainly gave the investigators enough support to move forward to a 2A study, or 2B study. One of the things that's of interest in using viral vectors, and particularly AAV1 subtype, is that I said it's a common cold associated or inducing virus. And you'd think that as you clear a cold, maybe potentially as much as years previously, that you would not have circulating antibodies in the circulation that are detectable, much less at very high titer. In fact, in clinical experience, many people have as much as a 50% of patients will have an antibody titer above a 1 to 4, in which if you gave this viral vector would inactivate the virus and the gene, and therefore limit the application of this therapy. It's as much as 50% of patients, and has been a problem in recruiting into these trials. The CUPID-2 trial now is into a second phase, the CUPID-2, which is 290 patients that have been enrolled using a recombinant. When the lesson was learned about the use of the adenoviral 1 as a vector, There was about 10 years of research that went on to use this non-replicating recombinant form, now referred to as AAV, adeno-associated virus. And unfortunately, the serotypes, one again has this high titer in humans. AAV 6 and 9 are other serotypes that actually are quite cardiotropic and relatively ideal, but the Food and Drug Administration of the United States has prohibited the company from just moving to a different serotype as the vector to use and have to go all the way back. So these trials are still being conducted with the adeno-associated virus 1 serotype. This trial has already gone to completion with enrollment, and it's an endpoint-driven trial, and it's expected that given the progression thus far, that this data should be completed by June of 2015 with ideally a late-breaking trial presentation at the American Heart Association a year from now. So this is a very important trial as a 2B. If it is positive, will lead to its progression into a phase 3 trial. The interesting thing about this is that when we talk about how you deliver this, this is a direct intracoronary infusion, and the method is a so-called stop flow, that you have a central lumen that you can continue to perfuse, and you can inflate a balloon for the segment in which you're actually extruding the vector and virus into the coronary circulation under some pressure, which has been shown to markedly enhance transfection of the gene. Interestingly, they chose to use a significant bolus of nitroglycerin intravenously to enhance viral uptake and delivery. So this trial will know a year from now the actual data and results, but a rather impressive early recruitment signifying the interest in this approach. And finally, in the Circuit 2A, one of the concepts discussed yesterday by Professor Yakub is the idea of recovery. And I think we're understanding a lot more of the molecular basis of recovery and heart failure. And the interesting approach here is in this phase 2A safety trial of using patients who have a left ventricular assist device implanted, both ischemic and non-ischemic etiology, placed at Harefield and Papworth in the UK. And the virus is infused 1 month post-LVAD, the same intracoronary delivery with stop flow and nitroglycerin pre-treatment. But I think this is a whole new area of investigation and use of gene therapy, is to be able to use this serially. Again, it's a human recombinant gene, so there really is no adverse effects, no rejection, no problems with its delivery, and using intracoronary approach doesn't have any impact on the LVAD and the continuous flow devices. So I think we're going to see, if this goes forward, a very significant impact in the field, potentially. A second gene that has drawn a lot of interest and clinical experience is Neuregulin. It's a very interesting and pleiotropic kind of gene in terms of its effects. It has definite effects in contractility, somewhat related to calcium handling, but also a direct effect in mobilizing stem cells. This has been in a phase 2B trial, which was stopped because a trial that was reported out of China showed a 24% survival increase. It's probably the first and only trial that has used survival as an endpoint. And so this gene was allowed to stop the 2B study, and they're about to start a phase 3 trial with this gene. It has somewhat unusual characteristics in that it has to be delivered over a time period of 8 hours so that you don't get peak titers. The adverse events have been pretty modest in terms of anticipating arrhythmic problems in its use, as we were part of the 2B study. But I think it's, as I said, going forward as a phase 3 trial that will probably be starting here in the next month or two. And again, it's just another virus that has been very successful. But the third gene that I want to really focus on is a gene called SDF, or stromal derived factor 1. Professor and I both, somewhat independently, have found this gene to be of particular interest when we were investigating clenbuterol, and found that one of the mechanisms by which this beta agonist was effective in enhancing cardiac function was it has a significant release of SDF. And this gene was able to achieve a lot of different effects in the myocardium, including enhancing stem cell mobilization. But It's very anti-inflammatory and has potent anti-apoptotic effects. So it is not a direct contractility or any calcium handling proteins, but has these unique effects that is an early response gene and theoretically a fairly critical gene in stem cell mobilization. This is the first trial of SDF in clinical medicine, and this is a graph showing this 40-patient trial in which a single administration was actually given transcoronary with a perforation into the coronary wall and delivered directly into the myocardium of a single dose of SDF within the first 2 days post-infarct. And what I've shown you here are 5 of the reported trials of using bone marrow-derived stem cells and the change in left ventricular ejection fraction. And I think you can see the average is about 2 to 3% in these randomized prospective trials. In comparison, for those patients that were treated with 1 single dose of the SDF gene post-MI who had an ejection fraction of less than 45%, the response was as high as 12.6% increase in ejection fraction. One of the problems in the field of both gene and stem cell therapy is that we've had relatively small sample sizes, and so we're getting a signal that may or may not be representative in larger powered trials, but a rather compelling difference in terms of a single dose of gene therapy versus stem cell therapy. And so I think this has led the investigators, Mark Penn, to move into a trial referred to as stop heart failure, in which there was a randomization 2 to 1 between 2 doses of the SDF gene versus placebo in a single dose. And the importance of this gene is that it's injected as a recombinant naked plasmid. It's not coupled to a vector and has really been an advance in the technology. This was now, like many of the new approaches with stem cells, was delivered into the myocardium through an endocardial catheter in the LV ventricle directly into the wall. And this biocardia helix catheter delivered these 15 injections without any perforations or complications and followed up. The 12-month data will be presented at the American Heart Association meeting next week in the response to this. Impressively, this has been very safe, and it is also, pending these results, going into a phase 3 trial of clinical investigation in January of this next year. But impressively, the investigators got involved with a new approach that I'll talk a little bit more later, which is, instead of using endocardial delivery, actually infuse it through the coronary sinus in a retrograde manner to get diffuse exposure into the myocardium. This is a trial of 60 patients that were enrolled in about a 3-month period with the exact same trial and using the same randomization of 2 doses of SDF. So we will have a side-by-side comparison of the benefit of intramyocardial versus retrograde delivery with this single gene to really get a clear assessment not only of the benefit of SDF, but importantly, the benefit of retrograde delivery. And I think given the simplicity of a venous injection, it's rather astounding that the coronary sinus, you can totally occlude it for a period of 60 minutes without any hemodynamic perturbations. And this is due to the thespian veins, which is essentially a pop-off of the coronary circulation to allow that blood to not cause any distension of the ventricle or hemodynamic alterations. So the wire is inserted, the catheter is inserted in the coronary sinus, a balloon is inflated on the catheter, and the gene is instilled over a 10-minute period, or cells in a similar manner, and allowed to dwell for 10 minutes, and then the catheter is removed. So it's a venous stick. It's much simpler, much easier to deliver. And so this is really a big milestone in the field of being able to have both retrograde and intramyocardial delivery of this gene. Professor has mentioned twice, and I just included to really emphasize this concept of, I think, is really rather important in the field, and that is the concept of this parabiosis, or cross-circulation of aged mice who had developed left ventricular hypertrophy and younger mice in a linked cross-circulation for a period of 6 weeks. And what was most impressive is that this exposure to young blood induced a regression of the left ventricular hypertrophy. But more importantly, perhaps, is that they look at the blood of these animals and were able to do genomic analysis and showed that there were 11 genes that were markedly upregulated in comparison to baseline in either of these animals, suggesting that there was a gene control of aging or regression of aging, and the most differentially regulated is this gene GDF11. I think anytime we see gene targets in a cause and effect relationship or a tantalizing hypothesis, potentially makes those gene targets that we can now take this technology of gene therapy and apply in that context. So this is going to be an area that's of great interest going forward as we really explore the concept of senescence and its control. So this is a look just in the area of heart failure, and these are genes that are right now in clinical trial. Again, this is hopefully going to go into a 2A trial a 3A trial from a year from now. Neuregulin is about to start its phase 3 trial. SDF is going to start its phase 3 trial in the second quarter of 2015. Adenyl cyclase I didn't mention, but beta receptor signaling is clearly another major pathway of contractility, and this is in a phase 2A trial now in the United States, looking at adenyl cyclase. Phospholamban is going to come right behind that as another look at calcium signaling. But I think that what's really interesting is the concept of, if you think that stem cells, particularly CD34 and endothelial derived stem cells, has now been coupled with endothelial nitric oxide into the cells. So we have a bone marrow derive the CD34 cells, transfect the nitric oxide within a period of hours to a day, and then reinstill these transfected CD34 cells post-MI. So we're going now to this next step in gene therapy, and that is coupling it to an ideal stem cell potentially. There's a lot of work going on with HIF-1 alpha and hemoxygenases, other ways to approach the acute MI story, but I think you get a sense that this is a field that is really rapidly accelerating, looking at specific target genes that make sense in heart failure. So I'm going to transition here kind of seamlessly now into the other half and much more data and support perhaps and experience, at least chronologically, in the realm of stem cell therapy for the treatment of heart failure. In a relatively simplistic way, the features that define a stem cell is that it has the ability to renew itself, and importantly that it has the ability to differentiate into different cell lines. One of the areas that has been a controversy is, should we have held any clinical trials until we knew actually the exact and precise mechanisms, and particularly the nuance of different cells, or should we get in the clinical arena and learn lessons in that regard? But I think when we look at the mechanisms that have been defined, particularly in the area of heart failure, there is very limited direct evidence of there being actual myogenesis, really creating brand new myocells from the cells that are transplanted. They may well induce the native repair mechanism and new myocytes, but as a direct cause and effect, there's very limited data. Some interesting data about direct myocyte division, but very little additional support. Without a doubt, most of the cells that we've been implanting, one of their key features is angiogenesis, which in the setting of ischemic heart failure and ischemic heart disease in general is very beneficial. They have a lot of anti-inflammatory, and as I suggested, much like SDFs, have some anti-apoptotic effects. But far and away, if you take away what is the mechanism that we think is playing a role in stem cell therapy, is probably the paracrine mechanism. And I come back to this same simple cartoon that whatever caused the injury to the myocardium and damage and ventricular dysfunction, when we transplant stem cells, they initiate this native repair, probably mediated in large part by genes to induce their improvement. And this is really the key two pieces of data, and that is particularly this work from Vic Zhao, which when he put stem cells in culture, the supernatant, when it was left for a period of 2 weeks, was drawn off just the supernatant, and it was equally effective as transplanting the cultured cells. And I think this really reflects the importance of this paracrine mechanism and what the cells are releasing. And secondarily, when you think of implanting millions of cells, you would say that they form a syncytium or develop this improvement based on that collective effect. But in fact, in most people's experience, certainly in the preclinical arena, the cells last for periods of hours to days, not months. And so the beneficial effect is not because the cells are there in situ, but they exert their effect and then are gone. The initial approach to using stem cells in cardiovascular disease was the leading cause of death at that time was acute myocardial infarction. We've had well over 10 years of using stem cells, and we have followed the natural approach at that time was largely using autologous bone marrow, with the concept that we'd done hematologic bone marrow transplants safely and had a lot of experience, and the thinking was that that's the only reservoir of stem cells in the body or where they're made. And so these patients underwent a bone marrow aspiration shortly after revascularization of their heart attack, and the cells were washed, and we've used both mononuclear fractions and specific subfractions like the CD34 cells, but they were washed and then instilled right down the coronary artery. And when you think about a bolus infusion without stop flow, it's rather interesting that these cells in fact do anything, but it's because the myocardial infarction caused the release of a number of cytokine and inflammatory signals which really serve as a homing signal for the cells to come to this area of injury. And so they were perhaps all of their effect was due to the presence of this homing signal. If you have an uninjured animal and you infuse those stem cells, about 70% of them will trap in the lung and 15% will go in the spleen and lymph nodes. And so it needs that signal to bring the cells to the anticipated or needed site of action. When you try to summarize in one slide 10 years of data, and there have been a lot of anecdotal series, but there are at least 17 randomized prospectively controlled trials, again, exclusively using autologous bone marrow. And I think overall, when you look at the benefit that you're looking at, it really hasn't been very robust. As I showed you the slide of those 5 trials, and again, it ranges from 0 to as high as 4%, somewhere in the range of 2 to 3% change in ejection fraction. That may not be the absolute endpoint of ideal endpoint, because follow-up of these patients in the German series suggested there's a longer-term 4 or 5 year difference in those who receive cells versus placebo. But in general, it hasn't been what we anticipated. Part of the problem has been that it's been an all-comer, and you realize in this new experience here of doing acute myocardial infarction revascularization, that the earlier you get to people in the United States, this has been in practice for many years now, that these patients are frequently revascularized and the stunned myocardium comes back, and many of these patients will go home from the hospital with modest, if any, reduction in ejection fraction. If you did a stem cell transplant on all those patients, you may not see much benefit if they were already above 50%. What we did find, though, that was those with the lowest ejection fraction had the greatest improvement, and again, this may be the relative concentration of the inflammatory signal that brought the stem cells into this benefit. This is a question about the benefit of stem cells in acute myocardial infarction therapy that should be answered fairly soon in terms of 2 or 3 years, because the BAMI trial is being conducted in Europe with a goal of 3,000 patients to really definitively answer this. And for the residents and trainees, this is a relatively good meta-analysis of the use of bone marrow for acute myocardial infarction. Well, the focus of today is in heart failure, and I think that the natural progression was to understand that we had developed catheter systems to be able to deliver the cells directly into the myocardial wall, not relying on intracoronary bolus infusion to try and deliver the cells to the myocardium, and do it in a targeted area that were all ischemics in the border zone of the infarct that we could map fairly precisely, and then deliver these cells for maximum benefit. This was a National Institutes of Health randomized placebo-controlled trial that was conducted several years ago. The patients were randomized 2 to 1 to receive this bone marrow mononuclear composite, versus placebo, and delivered 100 million of these cells. This was done by this NOGA catheter that I'll show you in just a moment. But I think the results were really very sobering, and that is when you did a subgroup analysis that was pre-specified, those patients who were over 62 years of age, on average, had no change in left ventricular ejection fraction, which was clearly one of the primary endpoints of this trial, trying to help people with LV dysfunction with these stem cells. And conversely, those patients in aggregate as a mean who were less than 62 years of age had nearly a 5% increase in ejection fraction, which by comparative standards to the MI was a fairly successful improvement. But it really raised the question of, is age going to be a significant limitation to the use of autologous bone marrow in particular, and really raised a significant question because age is where we see this is such an age-related disease. So we know the bone marrow is safe, but it may be an overall limitation. This data from Alan Kaplan, published several years ago, really perhaps gives us some insight into what's going on. This is looking at the relative numbers of mesenchymal stem cells in the marrow by age. And you can see in the newborn, 1 in 10,000 cells in the marrow are actually a primordial stem cell, but with a very rapid reduction in each decade of life to the point of 70 and 80-year-olds, maybe only 1 in 1 to 2 million cells. And so when we use the bone marrow in these older patients, we don't have much of a reservoir of actual stem cells. When you look at circulating levels of stem cells, it has this same direct reduction over time. And thirdly, when you look at work looking in patients with chronic renal failure and chronic lung disease, comorbidities dramatically reduce the functional capacity of the cells that are there, and their mobilization and migration is really reduced. And so when we use older people, particularly with chronic heart failure and chronic conditions, using their own autologous bone marrow cells probably has some real question marks about their efficacy. Well, that would seem to paint that picture as age is a barrier, but I show you another relatively small sample of 60 patients that was reported at the American College meeting last spring. Here, the average age is not atypical for those with ischemic heart failure, so 65, so they're older than was shown in the FOCUS trial and had a low ejection fraction. They again were randomized, but this time in the bone marrow, we're not using the aggregate bone marrow mononuclear cells, but looking specifically at bone marrow-derived autologous mesenchymal stem cells, subfraction out of the bone marrow, and again, the endocardial delivery. In contrast to what we saw with acute MI or what we saw in the FOCUS trial, which none of the endpoints were positive, this was really quite an impressive improvement. And the p-values, I'm sorry, seem to be moved off this slide, but all end systolic volume change, ejection fraction improvement, stroke volume, and end systolic mass estimated by MRI all had very significant improvements in response to these mesenchymal cells. And again, this concept of using MRI to quantitate the infarct size actually saw some reduction in comparison to placebo. And so it may be that bone marrow in general it may not be a very good source, but it may be that subfractions like the mesenchymal cell may point out the relative importance of that cell. And lastly, with regard to autologous bone marrow, again, the CD34 endothelial progenitor cell has been a target population of regenerating new vascularity into the myocardium. But this trial was really rather astounding for several reasons. This was a group of patients that were recruited back in 2005 and 6. There were 150 patients were screened and 131 were enrolled. And these are patients with angiographically defined non-ischemic cardiomyopathy. Again, a low ejection fraction, dilated ventricles, very high BNP. So this is a chronic non-ischemic heart failure population. Interestingly, nuclear scans suggested that there were target focal areas of reversible ischemia, even though angiographically, and the incidence of diabetes was rather modest. So they had G-CSF injected to try and mobilize a larger number of cells in the circulation in the bone marrow, and were given in a 1-to-1 randomization by intracoronary delivery. And so, as I said, these are chronic stable heart failure patients. You would think that endocardial delivery would not actually achieve a lot of local delivery into the myocardium to see a benefit. But the second part that's most important about this data is this is 5-year follow-up data of this fairly substantial sized population with non-ischemic heart failure. Those of you who have been involved in heart failure would recognize this as probably the most significant improvement in cardiac function, especially 5 years after a starting population with an ejection fraction of 25% and an LV dimension of 7 and a BNP level of 2,400. The ejection fraction average increase was 6%. 5 years later, it ranged as high as 12%. The 6-minute walk distance was 50% higher. The BNP reduction was an average of 54% lower than at baseline. Total mortality was significantly lower, pump failure death, sudden death, all reduced in response to one single intracoronary administration of CD34 cells. So we wouldn't perhaps believe, if you're trying to induce myogenesis and improve ventricular function, that a cell that is defined as an endothelial progenitor cell would have this benefit. But this is really compelling data that is now leading to other trials looking at the non-ischemic population. So when we've had, before this last data was released, there was so much concern about the autologous bone marrow as a logical source that it led to a lot of research into alternative sources for the cells, and this was potentially a switch to an allogenic source, looking at new types and sources, looking at cardiac progenitor cells and genetic engineering. If you think about the concept of organ transplantation, you're giving foreign tissue, which your body immediately recognizes as foreign based on surface antigens and markers, and you would require immunosuppression in today's world, 4 drugs to control your immune response to tolerate the presence of this foreign tissue. So the concept of doing allogenic stem cell transplantation seemed to be antithetical and not very good trade for the individual patient. But research has shown that the mesenchymal type stem cell is unique in that it does not seem to express co-stimulatory molecules on the surface, which are critical to mounting an immune response. And this cell is now referred to as immune privileged because in obviously outbred animal models, they showed no rejection, but it has gone on to clinical trials in which the assay of mounting an allo response is the presence of donor-specific antibodies in the recipient. The average prevalence is less than 5%. They're usually there for 4 to 5 weeks and then are dissipated entirely. And so as a single administration, we're now able to transplant allogenic mesenchymal cells. This has gone into extensive clinical trials. I would repeat that we don't have direct evidence that these cells differentiate into functioning myocytes, but they have had encouraging signals that they are changing ventricular function and enhancing myocardial function. We had, the first trial was done by Josh Hare, which showed that these allogenic cells were at least as good as autologous MSCs, and then a host of trials going forward that this phase 2 trial of this allogenic MSC has led to the development of the so-called DREAM trial, which now we're looking at trials of 1,700 patients as a phase 3, which if it's positive will lead to commercialization of these allogenic MSCs. One of the biggest benefit of these cells is that if we have an ability to select the donor, we're now taking a completely screened 20-year-old, essentially ideal health, no family history and so forth, as the single donor and just passaging that cell continually to have a single donor for all these cells, so there's no variability in the donor cells to see what the biologic effect was. But get a sense of how this field has progressed from small studies of 30 and 40 and 50 patients to enough basis to take a risk to do a 1,700 patient international trial, and we're actually enrolled in this trial right now and actively screening and enrolling patients. All-Star, we'll talk about a little bit. NIH LVAD trials are using these bone marrow derived mesenchymal cells in an allogenic source, and that's going to be one of the new trials using the LVAD again as an ideal platform, and it's also adipose derived cells will be used. Well, we began by talking about the bone marrow as the primary source of where all production is, and if you look at this field even 4 or 5 years ago, we would say, well, there's certainly an interest in embryonic cells, but the bone marrow is the single and only source of cells. But in today's world, as we look at it, it's very clear that there's a very robust repository of stem cells, surprisingly, in the adipose tissue, but also to get away from the controversy of embryonic tissue, the umbilical cord really represents both Wharton jelly and other preparations of the umbilical cord, and menstrual cells as well as the placenta. All of these have robust concentrations of not only endothelial progenitor cells but the MSC fraction, and when these are looked at side by side using bone marrow versus umbilical cord, Obviously, these are totally normal telomeres, and by functional assay are more robust MSCs than those derived from any other source. We'll talk a little bit about resident progenitor cells and then leave Professor the entire area of genetically engineered somatic cells. But I think you get a sense that the field has expanded rather dramatically in terms of sources of cells. One of the paradoxes that I have yet to come to understanding just teleologically in human development is why, at least by 3 different reports, that there are 500 to as many as 2,000 times more total stem cells in abdominal adipose tissue than there are in the bone marrow in an age comparative relationship. This is just that comparison of looking at bone marrow versus adipose tissue. The way these trials are done is that actually patients undergo a conventional abdominal liposuction, and the lipoaspirate is then put through a machine that simply adds the enzyme collagenase, which lyses away all of the fat cells, and you're really left with this pedicle of very concentrated stem cells. The adipose tissue is not a singular cell, but it has CD34 cells and a very high number of mesenchymal type stem cells. So it is more of a composite, like we said, bone marrow mononuclear cells, but this composite seems to have some benefit. Following very encouraging data about infarct size reduction and myocardial blood flow with the use of adipose-derived stem cells in acute MI, a trial called PRECISE was conducted in Europe and achieved a CE mark. When we looked at change in end diastolic volume, you see the natural history is for the ventricle to dilate over time, and this is a chronic ischemic heart failure population, not acute. And this is a single administration of adipose-derived regenerative cells by endomyocardial delivery in stable chronic heart failure patients. In contrast, when you look at the data at 6, 12, and here all the way out to 18 months, you can see that there was a fairly significant reduction in the natural remodeling that occurred. And this trial data has led to a trial ongoing in the United States right now called Athena, in which we're randomizing patients with ischemic heart failure to a single administration of adipose-derived regenerative cells. One of the holy grails I mentioned yesterday in terms of risk factor prognostication is the peak oxygen consumption, and using that as an endpoint in this initial Athena trial in Europe showed that in comparison in this one-to-one randomization between cells versus placebo, the placebo group were 8 years younger than those who received cells, and yet they showed the natural history over 6 to 18 months of a significant decline in their peak oxygen consumption, nearing the threshold for cardiac transplantation. In contrast, those patients who got a single administration of adipose-derived regenerative cells had a stabilization at the least, but impressively at 18 months showed quite a difference between the natural history of those untreated. And again, this was the foundation of moving to the Athena trial in the United States. This is a look at how these cells are delivered by 2 different systems. This endocardial catheter has a protrusion of only about a 4-millimeter needle, either by straight needle or this new corkscrew configuration, but can be injected in small aliquots in very targeted areas. And the precision with which we're able to map and deliver these cells now is within a millimeter, looking at animal models. And so this system has seemed to be quite safe, and steerable catheters with a lot of ability to put these cells anywhere that's targeted. And using this particular NOGA system is one other way of defining exactly the region that we're trying to administer the cells. The other source that I think has gained a lot of attention is the concept that potentially every organ in the body has a population of resident progenitor cells. When I suggested that gene therapy or even stem cells have this response to try and repair the myocardium, it may well be that there is a population of resident cells who are predetermined to only manufacture that lineage. When we transplant cells, as I said, one of the things that defines a stem cell is its ability to de-differentiate into different cell lines. When we think about being able to identify a lineage specific, like a cardiomyocyte cardiac stem progenitor cell, that has theoretical advantages. And all I can tell you is that it's been described now in brain, kidney, pancreas, lung, and liver. So the concept of a resident progenitor cell is gaining a little more foothold. There have been several cardiac progenitor cells identified, one by surface markers called c-KIT, one identified as cardiospheres, because in culture these things coalesce into cardiospheres, and there are several trials that have gone forward. They've had some controversy in interpreting, and again, they're relatively small sample size, but this one particularly using the cardiosphere, this is a 3-to-1 randomization dose ranging trial versus placebo. But importantly, I think when you start looking at the results and taken with the perspective of a relatively small trial, using MRI to quantitatively measure infarct size in grams showed that they reduce scar mass in association with delivering a single administration of these cells by endomyocardial delivery. There was some increase in overall mass, which has been interpreted by the investigators as suggesting myogenesis. That's relatively controversial. But they did reduce LV end-systolic and end-diastolic volume, which is the positive reverse remodeling as a goal, and the ejection fraction was improved rather substantially. This relatively small size and these results have led to the initiation of the All-Star trial, again a 2B, 274 patient trial. Interestingly, they changed from using autologous derived cardiospheres to now an allogenic source that also does not seem to induce an immune response. But these trials now are up to phase 2B. And perhaps lastly is the very exciting area of being able to transform by genetic engineering skin fibroblasts. And this is a look at the transcription factors that these cells were exposed to in vitro, and were able to differentiate, and Professor will talk about directly showing functioning cardiomyocytes, but that initial observation has been enhanced to now the concept of using this sequence of exposure to transcription factors to create these inducible pluripotent cells. This is one of the biggest advances and relatively astounding ability. One of the investigators in this field has been able to use a cocktail of 4 different factors that has been able to induce cardiac MSCs and gone into clinical trials in Europe. This initial pilot trial, you'll be interested to see in terms of results, also showed this reverse remodeling with reducing LV end-diastolic and end-systolic function, and also showed a reduced LV scar from infarct by MRI, and again increased mass. So almost superimposable results using this strategy and an increase in ejection fraction. This data in this 45 patients has led to the release of this Chart 1 phase 3 trial in Europe that now essentially is almost totally enrolled and has a 1-year endpoint. So we'll know fairly soon whether this strategy and these cells that are transformed will in fact be able to sustain these benefits that are seen. It is now about to enroll in the first quarter a phase 2B trial in the United States. One last thing I want to share with you is that when you read all of the results of stem cell therapy, particularly in the area of cardiovascular disease, it's important to understand that they are all based on a single administration. And as I say to patients, if you went to an oncologist and said, well, you have cancer and we have this regimen that we're going to use to try and help you, but that's it, there are no other strategies, or what if we gave you a second round or a third round and you see how chemotherapy is administered? Imagine if you could give a patient, when we look at the benefit, particularly in remodeling and ejection fraction, it plateaus typically at about 6 months. If we're using autologous cells or we're using inert, if you will, genes, why couldn't we see an additive, continually enhanced benefit of a second or a third administration, particularly as we get into a lot easier administrations? And so there is a trial of acute MI patients getting a repeat administration at 6 months that should be reported out next year, but that's only acute MI. We actually are conducting a trial off-site in the United States that looks like this, in which the patients are randomized to getting stem cells or placebo, and at 6 months you see a comparative difference in echo, 6-minute walk, and BNP levels, and then both groups, the placebo group has the benefit then of actually getting cells, so we can again see the benefit of cells, but importantly, the patients who got the first administration of stem cells will get a second course, and then we can begin to assess whether repeat administration has the potential benefit that we hope for. So when you look at finally, why would stem cell therapy gain traction and why is it more beneficial? I remind you that until this Novartis drug came in the field of heart failure, there has not been a drug that's been approved for clinical use for the last 14 years. And that really speaks to the concept about what we see as problems with drug therapy. And if you do a side-by-side comparison, and this is my own opinion, of stem cell and gene therapy versus oral drug therapy, at least thus far, we don't have any documented direct causal adverse effects from either stem cell or gene therapy to date. No hemodynamic changes. We don't know genomic predisposition to benefit, but this may be related in part to certainly the problems with metabolism of drugs. There is an age issue that we are certainly going after. We don't know there are any direct cause and effect long-term side effects, and the safety risk here is only during the time of administration. And remind you that when we talk about side effects, about 15% minimum of any drug that's gone into clinical testing has been discontinued for patient intolerance and side effects. And so as a comparison, these two agents seem to have a really fairly good profile and don't require the addition of another drug. And I think the scalability is certainly going to rival that. So I'll conclude with just a summary again of the stem cell trials that are in clinical trial right now in the United States and have already gone through trials in Europe of allogenic cells, bone marrow modified cells, progenitor cells, genetically engineered cells, adipose cells. We're beginning to start the umbilical cord trials with MSCs and then comparing them with the progenitor cells, and using the mechanical assist device as a platform for both allo MSCs and bone marrow derived MSCs. And similarly, the gene therapy trials that are actually up in clinical trial, and you see many of these are into the phase of 2B or 3, which means that if they're beneficial, will go into clinical application and availability. So a lot of progress being made in both stem cell and gene therapy. So in conclusion, I think that when you look at this field, you have to appreciate that most of the data are relatively small sample sizes, and so we're really getting a signal and trying to not overinterpret that response. The issue of age, I think, has a lot of focus on senescence and how we might be able to overcome that, and a lot of effort now directed at trying to retain these gene or stem cell products so that they may have an enhanced effect, better delivery systems, and I think that the concept of allogenic source of MSCs, because of the off-the-shelf availability and ease and ideal nature, seems to be an attractive hypothesis that, as I showed you, are getting extensive trial, and I think the genetic engineering will certainly be one of the most significant advances in the field. I think I'll stop there and hope that I've given you an overview of where the field is going. It's moving very rapidly. I would tell you that when you look at clinicaltrials.gov as a website, which records all the clinical trials going on in the United States in the area of just stem cell therapy, there are 4,500 clinical trials ongoing right now. This is a very broad application trying to harness and direct the body's native repair mechanism. Clearly there's a lot to be learned yet. A lot of that 4,500 trials are in the area of hematologic disorders, but a very broad overview, and hope that it gives you a sense of where we are and sets the stage for the discussion on the important genetic engineering.
Thank you very much indeed.